Okay, so without further ado, let's go ahead and get started. This is week 9 of our fall Bioinformatics Bootcamp. And just as a reminder, Bioinformatics Bootcamp is brought to you by the folks at Big Club. So if you have any interest in continuing to develop these skills and being more of a part of our bioinformatics community here, or at least the student bioinformatics community, feel free to come to our club and we'll be happy to talk bioinformatics with you. Okay, so hopefully everyone's downloaded this and opened up the script. One second. So what we're going to be doing this time is we're going to be talking about some of the public databases and public resources that are out there in order to get data and do some of these analyses. And this is really for convenience sake. At the end of the day, you don't actually need any of this to mine public RNA-Seq data, but it makes it a lot easier. And you know, sometimes that makes the difference between getting the result and not getting it. So I'm going to zoom in just a little bit to make it easier to see. So let's go ahead and load up our libraries. Hopefully everyone's following along with me here. You should all be pretty familiar with how we do these at this point. And of course, if you don't have any of these packages, do download them. But by now, everyone should probably have all 3 of these just because of the previous work that we've done. All right, so the first resource we're going to look at is Grind. Now, so the reason we're doing this, right? So what if you wanted to get RNA-Seq data, but Recount 2 didn't have the study you were looking for? It didn't have the kind of data you were looking for. Well, there's a few other resources you could go to. The first one I want to talk about is called Grind. And so let's go ahead and actually open that up in our browser. So it's right here. It's the GeoRNA-Seq Experiments Interactive Navigator. Basically, it's Recount 2, except different, and there's some different studies in it. So in most other ways, though, it's pretty much identical. And so what's really cool about this is that you can actually request new data sets be added into the database. So if something isn't in this database and you wanted it, you can actually send a request for them to add it in. They probably won't, just because as far as I can tell, they move very, very slowly. But it's kind of cool that they thought to add that feature. I don't know. At the end of the day, they really only have about 20,000 data sets, or sorry, they only have about 8,000 or so data sets in their database. And they're supposedly in progress processing another 20,000. So it's unlikely they'll get to your user requested data set, but you know, someday. Let's think about, though, what do they already have here? So I actually downloaded, I previously downloaded one of the studies from here. And in fact, if you look at the week 9 folder, you can even see it here. It's this GSE 101770 one. So if I search that here, yeah, there it is. So it's iPSCs. Oh, right. So this is how I got that initially. So if I unselect this, if I just search for iPSCs, I suppose this is probably one of the first ones that came up. And yeah, it's in here somewhere. The point is, just like with Recount 2, you can search using metadata to find any sort of study you're interested in, and then you can get the data from it. But I've actually already downloaded this data. So if you want to know how I got it, like I said, it was from that GSE 11070 study. Whoops, sorry about that. 101770. It was from this one. And all I did was I clicked on this. It gives you information about the study. So it gives you a summary of it, the title, and then it gives you some more metadata describing the samples. So these are your samples. This is the metadata describing them. So there's some hepatic organoids and some hepatoblasts and some iPSCs. And then you can also get the counts table. So this is just to show you how I got that counts table. If I want the raw counts, which I do, I can say show counts table and it will show it here. And you can just kind of see it nicely displayed. But really what I would want is to download it. So I download it like this, and that's how I got that CSV file. So now I've already provided this for you here. It's this one. So we can actually just go ahead and load that up into R using the read CSV command. And so I can see this here now. I've got my gene IDs, I've got my gene symbols, and I've got my read counts for each of my samples. So each one of these along the top of these GSMs, that's an RNA-Seq sample, and then the number of counts for each gene is displayed in the matrix. Now, if you'll recall, what we really need is we need this to be a matrix that has row names that are gene IDs, and then the only thing in the matrix should be the count data. So we need to get rid of this gene symbol row. We're actually not going to keep that. So we're going to get rid of it here. And then we're going to convert the X1 column, that's this column, we're going to convert that to the row names with column to row names. All right. Right. So I guess I should probably warn you about something that I just saw. You actually need to use dplyr select here because of an issue that I have not yet talked about called namespace. But just know for right now that if you don't do this dplyr .select here, it might take the select function from a different package. So for example, ENSDB H-Sapiens V86 also has a select function. So it was confused about which one I was talking about. So I told it I specifically want select from dplyr. And then it said, oh, okay, that's fine then. So that's how I get these tidy counts here like this. And so now I've got the row names are the gene IDs, the columns are the samples, and then each of the data points is the number of read counts for that gene in that sample. All right. So again, if you get that error, you can simply specify the namespace like this, and it will resolve it. Okay, so now to actually get our metadata, we want to use the metadata from the website. So we can see here, this is the metadata. It goes, organoid, organoid, cell line, organoid, organoid, cell line, tissue, tissue, cell line. And so I want to compare cell lines to tissues to organoids. And so because I know this order, I can simply put it into my data frame like this. So organoid, organoid, cell line, organoid, organoid, cell line, tissue, tissue, cell line. And that's going to be in the same order as this here. And I can actually confirm that after creating this like this. So I can confirm that. So it goes from 71 to 79. And then this went from 71 to 79. So it's the same order. And so my labels are the correct ones. So now I can, just like normal, I can build my DE-seq data set. Okay, so just for like a quick poll of the room, does anybody want to tell me what this error means? I mean, you can say it quite literally, but does someone want to take a guess as to what's causing this error? So Catherine says alphanumeric values. So if we recall, actually our tidy counts are they are numbers. So if we actually look, we can prove this. So if I do class of tidy counts 1, 1, it says it's a numeric. So it is actually numeric data. It's not strings. That's a good guess, though. That's pretty close, actually, to the issue that we're facing. It's just not strings that are the problem. Does anybody else have a guess? So, Simon, I think you accidentally. So, Dr. Chang, you're not supposed to. You're not supposed to guess. You know the answer. So I think, Simon, you actually meant to. Yeah. So yeah, that's right. So yeah. So Dr. Chang and Simon both said it. It's floats versus integers or doubles versus integers. Now, for reasons that I'm not going to go into, the statistical model that DGE uses only works on discrete data. So that's integers, right? And when we think about that, it kind of makes sense, right? Because you're supposed to have a certain number of read counts in a particular gene. So in this sample, I've got reads, right? Like maybe I've got 15 reads that align to this gene. Well, that should be 15 counts, not 15.1 or 15.2. That should be just 15. However, the issue is that sometimes you have a counting algorithm that doesn't use whole numbers because it's able to do sort of a multi-mapping approach, or I guess another way to say that is kind of like a fuzzy sort of mapping approach, where it can say, all right, maybe there was 15 here, but we're actually not super sure about a couple of them. So we're going to give half here and half somewhere else. And that's how you can get to decimals, even when you're doing the count data correctly, even when you're deriving it correctly. So to make DGE happy, the way that we deal with this, as wrong as this might feel, the way that we typically deal with this is simply to round it. And we can actually apply the round function. So if you recall, round is a function that if I give it, 1.0001222 something something, it'll round it to the nearest whole number. So basically what I'm saying is I want to take this round function and I want to apply it to everything in my matrix, to all the numbers in my matrix, right? And that's what I'm doing here with apply. So if you recall, apply is going to apply a function across a matrix, and it's going to do that either with respect to rows or columns or both. And so I'm saying apply it across both rows and columns. That basically means apply it to every cell of my matrix and then round them to the nearest whole number. And so that's what this is going to do. So it's going to make some rounded counts. And so now if we look at our rounded counts, we can see that they are in fact whole numbers, which is what DGE needs. And now it works. All right, does anybody have any questions on what I just did there? Okay, so once we do that, we can now take the R-log transform just to show that this data just kind of visually inspected. We can take this R-log transform, and once this finishes, we can do our plot PCA. And certainly not the most aesthetic data in the world. I would say that we probably would never want to actually go on to analyze this way, or we probably want to spend a little bit more time trying to understand what's causing these different groupings in the data set before we try to analyze this. Because it could be that this is batch effect. It could be a lot of things. We'd probably need to spend a little bit more time with it. But instead, right now, just for the sake of time, we're going to move on to our next data source. So instead of getting data from GRIND, we're actually going to try getting data from GDC, which is the Genomic Data Center, I believe. So let's go ahead here. Genomic Data Commons. Okay. Now, this is a really amazing resource, especially if you're a cancer researcher, because you can get all the data from TCGA. I think even targets in here. There's a lot of studies in here. And it's been standardized, so the data is comparable between studies, between patients, and there's a lot of clinical data here too. So when we do next semester, when we do our seminar on clinical omics, we'll be talking about this resource in addition to some others that provide a lot of similar benefits. But okay, so in this particular case, I'm just going to show you how I would get some data from this. And in fact, how I got the data that is currently sitting in your week 9 folder. So you can come over here to the repository. And let's say that I want to search for cases related to ACC, which I'm honestly blanking the name on the what ACC is. Let's see here. I think it was, there we go. Adenoid cystic carcinoma. So let's see if we can find that here. All right, well, apparently I don't remember where I got this from, so. Anyways, oh, yes, you're right, adrenocortical, yeah, okay. So this would be the adrenal gland then. And therefore, I have my, right, and then I want TCGA. So, okay, this is how I did it. So I said adrenal gland, and then TCGA. And then I can specify different conditions such as male or female. I can specify alive or dead, and so on and so forth. I'm just going to pick males for right now just to cut down on the amount of data I'm about to pull. Because at the end of the day, sometimes downsampling can make it a little bit easier to do the analysis in the first place, which is what ultimately we care about here. So let's get the RNA-Seq data, and then we're going to want the HT-Seq counts. So the difference between these counts is going to be your raw read counts. So that's the number of reads that align to any particular gene in each sample. FPKM is a type of normalized count, and then FPKM UQ is another normalization, which I'm personally just not familiar with. But we really don't want either of these when we're going to be doing our differential gene expression. And then we want the data in text format, but I think all the files probably already are in text format. And okay, and that's about it. So basically, it's showing you here how all of the filters that you've just applied rendered into a query. And so this is basically the query. So it's saying gender is male, and disease is adenomas and adenocarcinomas, and primary site is adrenal gland, and TCGA and HT-Seq counts, so on and so forth. And this is the search that we conducted to get all of the files that are the RNA-Seq read counts related to our disease that we're interested in here. So we can go ahead and add all of this to our cart. Now, you're not actually paying for this. Oh, apparently I've already done this. So it didn't add anything because all of these files are already in my cart. But for you, it will actually do that. And then now what we're going to want is we're going to download this data. So we can download the cart here. And it's going to prepare that and download it. Okay, so now that's done. Now we can download the sample sheet. And we can download the clinical data. That's what we want. Clinical data as a TSV file. And you're going to see in just a second why what we did makes sense and how we're going to use this. So if we come back over here, I've actually already downloaded these things. And in fact, if I show you in our week 9 folder, you can see those downloads are actually already in here. I just wanted to show you where did I get those from and how did I get those. So this is our sample sheet. This is our count data. And then this is the clinical data that we downloaded. Now, can anyone tell me what a .tar.gz file is? Okay, Simon says it's an archive. Yeah, It's, yeah, exactly. So it's like zip or rar. Yeah, that's really good. So .tar.gz, especially the .tar part is the archive part. .gz is a kind of compression. So it's compressed and it's also, yeah, so it's compressed and it's archived. And what we can do in R is we can actually unpack this archive. So we're going to do that here. So this command's going to untar the archive and put it into a new folder called GDC ACC cancer. So go ahead and run that. And you can see here now this new folder GDC ACC cancer has been created. And so it contains all of our 31 samples with their associated count data. So let's say here, this is one sample, and here is the count matrix from that sample. So this is akin to, if you recall, like our tidy counts, this is akin to one of the columns in our tidy counts matrix. It's just that it's in its own file here. So how are we going to get this count data and wrangle it so that it's ready for DE-seq? Well, first of all, let's actually get our sample sheet and our clinical data and just see what kind of samples we're working with here. So to get the clinical data, you're going to need to untar that file. So that was, that's this one here. So we're going to untar that one as well. And we're not actually going to assign an exit directory for it. We'll just let it be where we currently are. And so you'll see what I mean in a second by that. So it unpacked it right here. So it unpacked it into these three files: clinical TSV, exposure TSV, and family history TSV. So if I click on clinical, I can actually open this in Excel just to get, you know, a peek at what's going on here. So this is all the clinical metadata associated with my samples. So I can see the patient ID, I can see the project ID, I can see the age of the patient at diagnosis, I can see their ethnicity, I can see days to death, so how long before the patient died, I can see race. And I ended up deciding for this analysis, I wanted to focus on this AJCC pathological T, because this is a really great system for typing tumors, and based off of things like size and other, you know, pathological features. So I wanted to see for this cancer, how is T1 different from T4? Because I suspect that there are genes that define the difference between these two stages of the cancer. So I come over here, and the first thing I'm going to need to do is actually get these two sheets as objects in my R environment. So let's take a look at the clinical data. So I just saw this. This is what I saw before in Excel. And let's take a look at the sample sheet. So the sample sheet is a little bit different. It's got mostly information about the read count samples that I just downloaded. So let's take a look here at this first one, this 0197E35. This corresponds to A5, sorry, A51B7073, which I really wish they had picked more user-friendly identifiers, but I suppose they're very very easy to keep unique. So let's search this A51B thing. So I search that and I find it here. So here's this file. So this file is this one here. So I'm going to open the location of that file, and I can see that it's inside this folder, which is 0197E35A. And that's actually this file ID here. So what does that tell me? Well, if I go back to this, I can see that that tells me that my folders correspond to my file IDs. And then the files inside these folders correspond to the file names. So it's a bit convoluted, I'll grant you that. But at the end of the day, I figured out my structure, and now I know exactly how to find each count file based off of the information in this sample sheet. Now, how does this sample sheet relate to my clinical data? Well, both of them have a patient ID. So here's my patient ID, and then here's that same patient ID. So what can we use? We can use a join to join these two data frames into one. And that's what we're going to do here. We're going to do an inner join. So Simon asked a really good question, which is each count file contains counts from only one sample. Now that's a good question. It should be that HT-Seq only does one sample at a time, but we'll see that in just a second here. And in fact, if we want to, and I can show you this here, we can read that file into, right? So it's not like recount, because recount will get the whole study as one table, but HT-Seq is going to do just one sample at a time. So if we actually read one of those, we can do read TSV to look at one of those. All right. So we can see here just the beginning of one of those HT-Seq files. So we can see it's got genes and it's got counts. And so, yeah, only one column of counts, and that's because it's only doing one sample at a time. Okay, so we did our inner join, and now let's take a look at this. So we can see here now that we've got our file IDs, our file names, and we've got here our clinical data as well. So this is perfect. So we actually have all of our data as we expected it to be. Only one issue, which is that now, for some reason, it's duplicated. Every sample has now been duplicated. I suspect that's because there was a sort of one-to-many mapping in the clinical data, but that's okay. We'll be able to deal with that in just a second. So first of all, I want to select only the relevant columns. So I'm going to select sample ID, file name, and AJCC pathologic T. So that's my pathologic indicator for my tumors. And then I'm going to say distinct sample ID. So I'm saying I want to keep only the distinct sample IDs. So I'm getting rid of the duplicates. And then I'm going to rename that AJCC pathologic T column to condition. Let's take a look at that now. Sometimes you need to specify the namespace explicitly. So if you get this error, it's because you need to do that. Sorry about that. I wish I had spent more time talking about namespaces before now, because it does come up every once in a while. But basically, just know that every time you think you're using a function right, but then it complains about something, it's possible that the function has to be explicitly defined within the namespace you want, or else it might be used by another package just without you realizing it. So the select function is also found in another package, and so R doesn't know which package I'm saying use select from. So that's why we have to specify it with this namespace here. When you type the function name as you code, the little tooltip will tell you what package it will take it from, so you can oh, that's a really good point. So yeah, so Simon brings up a really good point, which is actually that RStudio will tell you as you're typing what package this is from. So yeah, so actually if I hit tab here, I can see that the version of select that it's choosing is from annotation DBI. That is not the one I want. I want the dplyr one. So now if I hit tab, it should say select from dplyr. Yeah. Yep. Okay, good. Yeah, good catch there. Okay, so let's go ahead and take a look at this. So I've got now my sample ID, my file name, and my condition. Perfect. This is exactly how it needs to be so that we can use the function DESeq dataset from HT-Seq count. This is really nice. This is a convenience function for DESeq where it can actually use the HT-Seq count output to automatically generate your DESeq dataset. as long as you give it a sample table like we've just constructed, the directory where to find the HT-seq output, which is, as you'll recall, that's this folder. So we give it that folder, and then the design, which is condition. But yet we find there is an error. Well, what's the error? Just out of curiosity, can anyone tell me why I might have gotten this error, or what this error might be telling us? Okay, so Simon's exactly right. It says it can't find the file, so it's probably a path issue. So let's take a look at the actual error text. Now, this is one of the most common errors you're going to get in R. And the reason is because it's just not super straightforward sometimes to specify where a file is to R. So whenever you get this error, what that basically means is that R was not able to find the file that you tried to make it find. And therefore, it says it can't open the connection. Cannot open file GDC ACC cancer something 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 HTC counts.gc. No such file or directory. Yeah, exactly. It can't find the file. Well, why can't it? Well, let's think about it. So if we actually go back here, we can see here that this folder, this GDC AC cancer, is not actually where those files are. The .gz files are not inside of this folder. We have to go one layer further to find the files. But in my sample table, I never told it that. I never told it how to find the files by going like a layer further. So like it's basically going to look for this file inside the parent directory, which is this directory. So what do I need to do? Well, there's a few ways I can handle this, but the easiest one, if we recall, is probably just to tell it where this folder is. Because remember, this is the folder that corresponds to this file. So this folder corresponds to this file. So if I go inside this folder, I'll find this file. So what I could do is in the file name, I could actually give it this folder / this file. And then it'll be able to find the file within that folder inside my larger parent directory. So that's what we do here. Basically, we're going to do this mutate statement. We're saying file name is now going to be the paste of file ID / file name. And so that's going to make our directory / our file name as the new file name. And it's going to complain at me again, so I'm just going to preempt this here. All right, so now let's take a look. Okay, good. So if we does not apparently like me doing that. All right, so now if we very carefully move this, we can see now our file names have the directory / the file name. So now DESeq should be able to find this. And yeah, it hasn't complained at me, so I guess it's happy. Now, it's still going to throw a little bit of an error, or not an error, but a warning at you. Basically, it's complaining that you used some special character that it doesn't approve of. And we're going to see in a second, actually, what we did. So let's go ahead and run our VST. So that's our VST transform, which is similar in many ways to RLog, but it's a lot faster and probably good for this sort of patient data. And let's go ahead and plot that. Okay, perfect. So now we can see our Pca plot, and we can see our T1 is kind of over here. Our T2 is sort of all over the place. T3 kind of all over the place. And then T4 is mostly on this side. And then we actually had a couple samples that had this weird sort of blank or null name. Maybe that's because I didn't know the classification for those samples. And this is probably what R was complaining about when it said levels of factors shouldn't have special characters. It was probably complaining about this. So that's where that came from. So just in interpreting this Pca plot, does anybody see I guess any issues, or like what's everyone's first impression of this? Is there anything we can really do with this? Or I mean, how would you consider parsing this? Yeah, so Simon says don't compare T2 to anything. Yeah, I would probably agree with that. Yeah, so I would say that one of the things that we could do here, if we really wanted to know, yeah, so Catherine and Simon both said T1 versus T4 is probably a decent comparison. And I would agree with you. In fact, I would say T1 versus T4 is probably the starkest difference I see here. And that's nice, actually, because those are the most extremely different kinds of tumors. And so that allows us to really see, well, what are the genes that most account for this sort of difference here? The other thing we could actually do that I'm not going to go into, but is totally worth looking into, is that you can actually use the likelihood ratio test to see what is the continuous change in gene expression between T1 and T4. So you could actually consider this to be a continuous sort of transition here. And then you could see what are the genes that are changing along that transition. We don't really need to do that for right now. Those can be a little bit harder to interpret, and it's probably not necessary since we can make a nice stark comparison between T4 and T1. But just so you know, that is something you can do. So as Muku pointed out, PC1 is really not ideal. It's 19% of the variance in the data. Yeah, and like you're saying, like you really cannot tell much from that. But because we're going to do a direct comparison of T4 to T1, that should afford us just enough sensitivity to see what are the most differentially expressed genes between these. And again, this is patient data. This is not uncommon for patient data to see sort of a weak biological difference between your samples. But anyways, let's go ahead and run it. And again, it's going to complain at me for a little bit because I have this weird special character group here. Okay. Oh, no. You got to be kidding me. No. All right. Sorry about that. Hopefully yours is still running. Oh, I have not had that happen to me before. It's the most disappointing thing ever. All right. Let me just try this again. Yeah. Did anyone else have that? Okay, that's good. I literally just had this working. That's crazy. BSOD. I'm actually sure what that means. Blue screen of death. Oh, yeah, no, it's kind of, yeah. Yeah, so Holly said that's happened before. Yidong said that happens all the time. Yeah. I guess I don't really, usually when that happens to me, I know why it happened, and it was because I did something dumb that I knew about. But this time, I don't even know what I did wrong. So, all right, let's go ahead, and sorry about that. Hopefully it will have calmed down this time. All right. If it crashes again, I don't know what to do. I guess you do know this part is really just stuff we've already talked about before. So this is where you're getting your mapping between the gene IDs and the gene symbols. And this is where you're actually getting your overexpressed genes. So, okay, it looks like it's not going to crash this time, which is nice. Yeah, I guess it's like it. I don't know what the actual computer science term for that is, but it's like it usually says core aborted or like seg fault or something like that. Not really sure why it happens though. All right, so anyways, it's running. It's continuing to complain at me that I had special characters in my factor levels. But, it's not really going to cause any issues. So while it's running, does anybody have any questions about what we've talked about so far or what I've shown you guys? Okay, well, guess it's just gonna be slow. This was so much faster earlier. Oh, okay, now it's going. All right, good. Okay, good. So now it's finally done. So I'm going to get my results, and I'm going to set my contrast as T1 versus T4 in the condition column. So basically T4 and T1 are in this column, and I'm saying in this column, compare T4 to T1, right? So that's how I specify my contrast. And so now if I take a look here at my results, I can see, if I sort by p-value, I can see this more clearly, but I can see what were the most, differentially expressed genes. I can just, come over here and I can Google this one, and I'll find it's it's ZIC1. That's super interesting. That actually is interesting for a few reasons, but basically what I want to do though is I want to actually convert those gene IDs to gene symbols, because at the end of the day, Googling one of these at a time is not really very efficient. So let's go ahead and convert them using the same code that we showed last week. All right, so now we've got our gene IDs and we've got our gene symbols. Perfect. So if I sort by p-value, then I can now see what are the top hits. Okay. All right, so let's say that I want to actually save the overexpressed genes to a CSV file. I'm going to show you in a second how we can use this, but let's go ahead and do that. So we want the p adjusted to be less than 0.05 and the log 2-fold change to be greater than 2. We'll write that to a CSV file. So we can see that here. That should be this T4 versus T1 overexpressed.csv. And great. So I'm going to take these genes and I'm going to copy them because I want to do my enrichment analysis. Now, we talked before about how to use Enricher for this. Now, Enricher is a great tool. I can paste this in here, submit the job, and immediately I can see what are the overexpressed pathways that are in my overexpressed genes. So basically, what are the processes that are being upregulated in T4 compared to T1? So some pretty interesting stuff here. But what if I want to do this in a different way? Well, there's a really interesting way that I can do this while incorporating another kind of data called protein-protein interaction data. So just to start off with, this is string DB. And there's a lot of things you can do with string DB. And actually, I'll just show you the most simple way to use string DB. Let's say that I want to look at, sure, I'll look at SLC 7811. So let's say I want to look at SLC 7811. I can search this. Sure. And now I can see the protein interactome for SLC 7811. So its strongest connection in the network of protein interactors is SLC 3A2. Some of you might know that SLC 3A2, also called XCT, is in a protein complex with SLC 7811. Together, they have an antiporter channel that is implicated in a lot of aspects of metastasis, actually. So this is really cool. I can see the whole network of proteins that interact with SLC 7811. And then if I look at the connections, I can see what is that interaction. So it's co-expressed. There's biochemical data that says that they physically interact. So that would be like affinity capture mass spec. That would be like two hybrid. So there's also curated databases that show that they're interactors, and they're also co-mentioned in PubMed abstracts. So a lot of evidence from different sources say that these two proteins definitely interact. Now we can also come to the analysis and we can see what is this network doing? What is it involved in? So here we can see one of our enriched pathways is positive regulation of transcription from RNA-Pol2 in response to oxidative stress. Okay, cool. Well, I know that NRF2, which is this protein, is involved in responding to oxidative stress. And this is one of the targets that it upregulates. So that makes sense that this network is also involved in that. Okay, glutamine transmembrane transporter activity. So I also know that a lot of this is involved in the production of glutathione, and so that makes sense. So a lot of stuff makes sense here. So that's the power of using protein interactors. Now, we can now take our gene list from earlier, our overexpressed genes, and we can actually paste that into here. So if you recall, what I did was I took this list of genes that are overexpressed in T4 compared to T1, and I just pasted that here. And just for the sake of time, I'm going to tell it that it's Homo sapiens, so it doesn't have to try to figure that out. And I can search this now. So it's going to map that. It's going to then do the analysis. And it's going to complain that you gave it a lot of proteins, and it doesn't like that. But it's OK. It's just going to hide a little bit of data so that it doesn't slow down my browser. All right. So now we can see the whole network. I'm going to zoom out just a little bit here. We can see the whole network now. Maybe that was too much. And what's really cool about this view is that we can actually see that there are clusters of interacting genes within my list of overexpressed genes. So this is really, really powerful for identifying not just what individual genes are overexpressed, but what processes and what gene groups do they belong to. And this in particular is a really cool finding, because if we start to look at some of these, we can see, CDCs, we can see CDKs, we can see SEN-PM, and then we can see histones. So what does this tell us? Does anybody have a just like if I zoom in here, anyone who maybe works in cancer or in molecular biology have a guess as to what this cluster might be involved in? Yeah, all right. So okay, I guess you knew I was going to ask. Yeah, cell division. Exactly, cell division. So yeah, so that's really interesting that we would see, replication, cell division. It's really interesting that we would see this really big cluster in our overexpressed genes, considering that we're comparing two different stages for the tumor, right? So what's also really exciting is that there are some other subclusters that have some significance. So I'm going to focus on this one over here. So over here, I can see GATA4, SOX1, PAU4, F1, EN2, EN1, FOXA2. So these are, if some of you are familiar with more, the developmental biology side, you might recognize some of these as being involved in differentiation and in lineage commitment. So we can confirm that by coming down here to the bottom where we did our analysis, and we can see that, in fact, a lot of our pathways are related to what we just basically talked about. So cell fate specification, neuron fate specification, embryonic skeletal joint morphogenesis. And if we actually click on this one, we can see here, we can see those, those proteins are lighting up. So it's kind of nice. You can actually compare multiple subpopulations using that. So hopefully you can see why I love strings so much, and I use it. Despite being able to code in R, I probably use this, at least a couple times a week, because I find this view of the data to just be very, very easy to interpret and very nice for actually getting to the bottom of what's going on with differential gene expression. Okay, so why is this cool? Well, because the cancer that we were talking about is an adrenal cancer, and neuronal differentiation is actually a really important component of that cancer and the progression of that cancer. So the fact that we see that in our T4 compared to our T1 actually tells us something really interesting and important about how this cancer works in the first place. Okay, one final thing that you can do with this before we get to some of the student presentations and before we talk about next steps. So one final thing you can do with this is you can actually do your gene set enrichment analysis. Now, as far as I know, there's no other places online where you could do a GSEA in your browser. There's probably some out there, I just don't know where they are, but this is one of them. So what we're going to do is we're going to make our GSEA metric like we did before. So it's a negative log 10 of the P adjusted value times the sine of the log 2 fold change. And then we'll write that to a new file, T4 versus T1 GSEA ranks. So let's go ahead and write that. All right, so here's my ranks. And this is actually perfect because what we need is we need the symbols on one side and we need the values on the other side. So we're going to take both of these columns, get them, copy, and we're actually going to go back to string DB, go here, and now that we're going to come to this section protein with values / ranks. So that's where we're going to paste this. It's going to take just a second. All right, so it's pasted all that. We'll choose Homo sapiens again, just because that's going to save us some time here. And then we go ahead and search. Now, this part's really going to take a minute. And the reason is because it's got to search through this entire... Oh, okay, sorry. First things first, it's going to find the NAs. And it's basically going to remove the NAs. But it's first going to ask you, do you really want me to do that? And you can just say, sure, that's fine. Okay, so now what's got to do is it's got to search through all of your genes and then map those to genes that it has in its database. So that's what it's doing here. So it's just making sure that it understands how do your genes relate to its database. And it should just take a second. Okay, great. So it's showing you how your genes relate to the genes it knows about. And then you can actually download the mapping between those two. There's really no reason to do that. I mean, so at some point you might want to, but for the most part, it's not really super important for right now. So we're just going to continue. And now it's going to actually run the analysis. So it doesn't strictly speaking do GSEA. It's actually got a newer algorithm that's very, very similar, but not quite the same. Dr. Chen or Dr. Zheng, you might be familiar with it. They use these things called AFC tests. But yeah, I'm just not familiar with exactly how it's different. I just know that in general, because it's still using the Kolmogorov-Smirnov, I said it right this time, test, it's actually essentially doing the same thing.
Yeah, this time you said it right.
Thank you. I've been practicing. So this is really cool. I actually, to be honest with you, I use this quite often because sometimes it's just kind of a pain to do the whole GSEA in R. And this you can just do in your browser really easy. Okay, so you can see here we've got our genes and the values. So this is our top underexpressed, FAM 17.8b. And then our top overexpressed is down here at the bottom. Is this ZIC1. Okay, great. And we can see the distribution represented like this. So we can see that in general, we had a good number of significantly overexpressed genes, but really not a whole lot of significantly underexpressed ones. And actually, their algorithm attempts to account for that in some way. Although, again, I'm really not sure on the math side how it's working. All right, so we can sort by top of input or bottom. So top is up here, that's what's underexpressed. Bottom is what's overexpressed. So I care about the overexpressed. So I'm gonna come over here and do that. So DNA packing is probably our top hit here for biological processes. So we can click that and see where that is. And we can see the gene networks that fall out of that. Chromatin assembly, nucleosome assembly. So these are all those histones we saw earlier. And I suspect cell cycle is probably, so kinetochore, so that's going to be, yeah, replication, cell cycle, ribonucleoprotein complex localization, and RNA processing. So that's kind of interesting. That's now something that we didn't see before, but which is also interesting to us. And then we've even got a little bit of neuron fate commitment down here, and just a few other things that are kind of interesting. So if we look at the other side now, if we look at the top of the input, so what's being underexpressed, we can see the top hit is this vacuolar transport, which I'm really just not really sure what part of the biology that relates to. I suspect someone who studies this cancer might have a better idea. But yeah, there you have it. So that's in essence the biological interpretation you can do with string, without ever having to actually do the GSEA or the overrepresentation analysis yourself in R. So this is just another way to do it. Okay, and the last part here, I wanted to just briefly show some of the other places you can get data from. And then this is going to tie into what we're going to do next time. And so this should just take a minute. So one of the places is Synapse. I actually have worked quite extensively with the data from Synapse because I've worked with one of the Alzheimer's patient studies. But this is typically where you can get a lot of really great curated patient data from. We're not really going to focus on it for right now because basically everything is hidden behind a data access agreement that you have to fill out and get Utasca to sign before you have any chance of getting the data. So unless you've got a really good reason to need this, you're probably never going to use it. Okay, next one, Encode. So Encode is really awesome. They've got some really great standardized RNA-Seq data. Although in general, they're typically more useful for epigenome sort of data like ChIP-seq and GROW sequencing and that sort of thing. So you can use the data from here, although it's a bit harder to use than it would be to go to another source like GRINE or ArchS4. And speaking of which, ArchS4 is the next one. So you can go to ArchS4. They've actually, this is all RNA-seq data. They've standardized literally more than 500,000 samples at this point. I mean, it's really incredible what they've done here. The only issue with it is it's a bit of a pain to actually find the study you're looking for. So for example, if I was looking for, Ewing sarcoma, which I can do here, Ewing sarcoma, I can search in here for this, and I can find that there's actually 59 samples associated with that and 5 studies. So I don't know which one comes from which, and I'd have to download it and parse it myself to really figure that out, but it's certainly doable. It certainly beats reprocessing all the data by myself. The only issue is that this download button does not always work. And in fact, I tried this earlier today and it did not work for me. So their actual download function is a little finicky. They do give you this R script that they automatically generate. And actually, I'm going to open this up so you can see. They give you this R script that if you just run this script, it will actually get the data for you. And it will write it to a TSV file. But to actually use this script, you have to download the entirety of their database, which is fine. Like if you're doing this a lot, then, it's fine. It's, several gigabytes. I think it's something like 40 gigabytes at this point. And if you've got the space for it, it's a good investment to just go ahead and download it, especially if you're going to use this service a lot. Like I have it on my computer because I use this a lot. But if you don't use it a lot, it's definitely a pain to download it once just for one study. So anyways, yeah, as you can see, this is really not working anymore. So that's ArchS4, which is probably a really great source as long as you're willing to download a 50 gigabyte file to use it. And then finally, we come to the largest, by far the largest source out there, which is GEO and SRA. GEO is, that's where the process data is. SRA is where the raw data is. But these databases are actually linked together. So let's go to GEO. GEO is where the process data is, and that's typically where you're going to search for data. So we can come over here. And so sorry, I guess I'm going a little bit fast. We can come over here to query and browse and do search GEO datasets. So let's say I wanted to find a study relating to Ewing sarcoma. I'm actually going to do this in incognito browser so it doesn't remember what I did before, because I'm sure I've already done this before. So let's search Ewing sarcoma. So I'm going to search for some Ewing sarcoma studies. And actually, I find a few here. But really, I want RNA-seq. So I'm going to select Customize under the study type. And I'm going to select Expression Profiling by High Throughput Sequencing, because that's kind of a fancy way of saying RNA-seq. So select that. And now I've got 60 studies remaining, and select humans. I don't want zebrafish. I'm not sure how that got mixed in here, but yeah, so this just takes a second. All right, so here's all the studies I can get data from. So for example, if I want to get data from this study, this Lin28B underlies pathogenesis and therapeutic sensitivity in a subclass of Ewing sarcoma, I can just click this, and then this is the information about the study. So here's a title, summary, design, and then I can even read the paper that came out of this study here. And then I can see the different samples that are available. So I can see, you know, EWS1 through 4, you know, same conditions, but with shRNA for Lin28B, different shRNAs, different inhibitors. And so what do I want? Well, I want the count data. I want the read counts from this. So what am I going to do? Well, the thing about Geo is that sometimes the data is what you want it to be, and sometimes it's not. But all the data is going to be pretty much down here. And if you download this, which I've actually already done, if you download this, you can get that tar file. So as Simon pointed out earlier, tar is an archive. So if we come back to, no, I won't make you do this. If you go to R and you untar that, you'll actually see, yeah, all right, let's do it. I don't know, why not? So let's actually untar that. This will be nice practice, I guess. So let's go ahead and untar that tar file, because I actually included that in here. So that's this 321 here, right? Yeah, so that's this here. So let's go ahead and untar that, and we'll make the exdir. We'll just call this this. All right, so now that made, whoops, I keep jumping back and forth. That made this folder here. And we can see all of our samples here. So that's great. That's exactly what we wanted. But there's only one problem now. Well, we've got all of our samples. And this is like we had before with our HT-seq. We've got all of our samples, and they've got the counts. But are they actually counts or not? Are they actually read counts, or are they something else? So we see this thing here. It says .tpm.tsv.gz. So I know we haven't talked about different kinds of normalization approaches for RNA-seq, really. But TPM is a normalization approach that some people like to take with RNA-seq. And unfortunately, TPM is not something that you can use for differential gene expression. So we're starting to get a little bit nervous here, because like, what if this is TPM? Then how am I supposed to use this data? Well, to verify that it's TPM or not, we can come over here and we can click on one of our samples, and we can go down here to the bottom and read the data processing statements. So it said they used RSEM to align the reads to the human transcriptome. And then it says supplementary file format and content. It's a tab delimited file containing the TPM of each gene. So it is the TPM. So we can't use this. That's it. That's pretty much all you get. Or it would be if there wasn't one last thing that we could do. Now, this is a bit of a last resort, but it's the one that, to be honest with you, if you do a lot of data mining, you'll find yourself taking most of the time. So the last resort is to go and get the raw data in SRA. So it's over here. You can go click this, and it'll take you to the SRA page. And if you click on any of these, actually, these are all the samples from the study, and then you hit this all runs, you can see the table containing all the data. So this is great. So we can see here our different samples. We can see the geo accessions associated with each one. So each of these is related to one of the samples in geo. And so there's a one-to-one mapping between them. And then we can see the conditions. So shRNA, no shRNA, Lin28B, GFP, and so on and so forth. So this is great. And all we need to do now is we need to take this data and we need to somehow get read counts from it. So if I come over here to run, you can actually see the raw data. And I'm not really gonna download it just because it's huge, but let's take a look here at the reads. So here are the reads. So you can see there's about 2.7 million reads. And here's one of them. So here's one of my reads. It's got my C's and my A's and my T's and my G's. And I can see, yeah, this is the first one out of the 2.7 million. So what would I do? Would I actually download this data? No, probably not. There's a more intelligent way to access all of this. But just know that that data is here. Everything that you find in the Geo database, every study that you ever come across in Geo, especially the sequencing studies, all of them are going to have an SRA accession. That's what they're called, accessions, where you can get the raw data. So no matter what, you can always get the raw data. And for the last part of our workshop, we'll be telling you exactly how to do that, how to process it, and how to get it to the point where you have, you know, something that's a bit more like, you know, a bit more like this. So we'll tell you how to get there starting next time. Okay, so just as a reminder, the homework for next time is to do the Introduction to Shell course on DataCamp. So just to make sure that we show that here, if you go to DataCamp, if you remember DataCamp at this point, I know it's been a little while, but if you go here to the assignments, that should be, oh, that's not what I meant to do. Oh yeah, so that should be there. The Introduction to Shell should be here somewhere. You can even just search it, just Introduction to Shell, and it'll show up. So do this before next time, because next time we're going to be working in Linux, and you're going to need to know a little bit of the basics for Shell. We are going to cover them next time, but it's just really good to have a little bit of a background before going in, simply because that will make it much easier to follow when we're actually doing it interactively. And as a reminder, if you have not done this yet, please do go fill out the form, register for next session so that we know whether or not you're going to need a virtual machine. So, and I'm going to share this in a chat in just a second, the Google form for that. But if you haven't done that, please go do that. We need to know who is actually planning to participate in those sessions so we can set up a virtual machine for you. Okay, so with our last 17-ish minutes, Simon has volunteered to present some of the analysis that he's done. So Simon, would you like to go ahead and get started?
Sure. So I'll just basically walk through my script.
Sure, go for it.
Okay. Screen share. There we go. Okay. So this is kind of a combined script for the homeworks of weeks 7 and 8 with the differential gene expression analysis and then overrepresentation and GSEA. I also included like Dr. Bonek's like visualizations. So yeah, I ran the whole script already, so all my plots are already here, so we don't have to go through that. So I went and found a study on Parkinson's disease on recount. It's a clinical study, and that will become painfully obvious once we get to the PCA. But basically, I did what Henry did in his code, where I, if we look at row names, well, it'll be fixed already. But all the ensemble IDs had like a dot and then a version number after them. So this line of code just cleans them up so that the names of the ensemble IDs are what's going to be in the annotation database. Let's actually see if I can. There's a ton of results here. Yeah, so that they look like this, basically. This is my results table, but whatever. Originally, they all had like little version numbers attached to them. So then I looked at the column data. Suppose did that now. So there's the column data. There's 73 samples, and there's 22 columns worth of information about them. And I honestly don't know what most of them are. All I know is the one column I needed wasn't there, namely which ones are controls and which ones are Parkinson's disease. So I went and like looked that up on the on SRA. I went and looked at what patients were what, or I think I looked at the actual paper. They had like a table on there or something. I forget where exactly I got the info from. But then I found that if you look at the names here, title is like the name. So this one is P_0019, and there's like a C_0081. So I figured out that anything with a P is Parkinson's and anything with a C is a control. And so here I used regex, like regular expressions, to go in and basically replace anything that has a P in it with Parkinson's disease and anything with a C with control, and make that into a new column called condition that you see here. So I should probably check it. I can't see the chat right now, so if anyone has any questions, just I suppose unmute yourself. I went ahead and made the DSeq object, and I specified that I wanted to compare control and PD. Then I did what Dr. Bunnick did. I did the Poisson distance between all of my samples. So this is how similar the samples are at the raw counts level to each other. And this magical graph got made based on it. And so you can already see the glory that is clinical data, because the controls and PDs do not cluster together as nicely as they would if you were, say, a cell line or something like that, where you just add like an shRNA or something and then did RNA-Seq on those. But, you know, they don't like perfectly cluster together, but they kind of sort of group together, which is, I mean, it is what it is. So that's this part here, is calculating Poisson distance. Just wrangling that a bit, taking the actual distances out of the object that gets generated here, turning it into a matrix, naming the rows, and then making a heat map with that blue color palette. I then normalized the data and plotted the PCA. So that's what the PCA looks like. Again, clinical data is very variable. This isn't like a inbred mouse model or a cell line where one is like a Parkinson's model versus control. This is humans that are like messy. And so most PD cases are in fact sporadic. So they don't have a nice little mutation that's causal. So there's just like a lot of variability. So the groups don't cluster that nicely. But you can see that they like, the preponderance of controls is on the left, and the preponderance of PDs is on the right. They just don't separate nicely. So then I ran the DSeq analysis and stored the results with a p-value cutoff of 0.05 and plotted. I don't think we've actually gone over this plot in class. This was on data camps like DSeq 2 class. So this is for each gene, what the statistical model did to it. So basically it like fits, it kind of adjusts the model for each gene based on the information from the entire data set that you give it. And I'm really not sure about how the math works there. That is way above my pay grade. But basically this red line is kind of where the fitted data falls, and for reasons that I cannot explain to you, this is the shape you're looking for. That it kind of flattens off, that the dispersion, which is kind of a not exactly variance, but a sort of a measure of variability, that it kind of flattens off as you increase the mean of normalized counts. Henry, you'll interrupt me if I'm saying something that's completely ridiculous, I hope.
I mean, to be honest with you, I'm not even super familiar with how, I understand that like they're trying to model the mean variance trend and then fit the data towards it, but I'm not, beyond that, I couldn't explain what they're really doing.
Yeah, samesies. But it's a very pretty looking plot, so I did it. And basically, like, if it sort of kind of looks like this, was my takeaway from the DataCamp class, you're fine. If there's like weird stuff going on, where there's like clouds of data just like sitting in the middle of nowhere, that there's probably something going on that you should investigate. Okay, next I did the MA plot on the not normalized data. Oh, I shouldn't have executed that. Yeah, go away. Oh no, I'm gonna have to click back through all of my plots now. Give me a minute, my computer will deal with it. We'll get there. Oh, so close. Is it done? Sweet. Okay. So I did the MA plot where you plot the mean of normalized counts versus the log fold change for each gene. So each dot here is a gene, and on the x-axis it's the mean of the normalized counts between all samples for that gene, and on the y-axis is the log fold change for that gene between PD and control. The blue ones are the ones that are significant. So you see that you have this like these dots here that are outside of the range that have like very large fold changes, but are still not significant, and that's because low fold changes can get overestimated, or like fold changes for genes with low counts can get overestimated by the model, and that's why we do this like log fold change, LFC shrink, that shrinks the log fold changes based on one of three mathematical models that you can choose. There's the normal, which will spit out data that is Gaussian, so it's normally distributed, or the Asher or the APE GLM. So I don't know enough about the math behind them to be able to tell you when you should pick which, so I just ran all three of them. So again, this is without shrinking. So if we use the normal, it'll shrink it like this. You'll see that all of the dots are way out of range or gone. If we use, excuse me, Asher, that's kind of what it looks like, which is quite nice and close to the axis here. But then again, you have these like high outliers again. So I don't really like this one for this data set. And then this one just looked weird, so I skip. So if I was gonna show someone something in lab meeting, it'd probably be the normalized one. So then I did 2 histograms of the p-values for either all genes or the ones that just have a base mean expression of bigger than 1. And basically what you can see is this peak here goes away if I limit myself to plotting only the p-values of genes that have a base mean expression of greater than 1. So that's probably genes that are so low expressed that you can't really say anything about them. And then I did a volcano plot. That will surely show up soon. So I didn't really play with these cutoffs too much because honestly, like I did not see, if I open this up bigger. Yeah, I didn't really see any populations that distinguish themselves that would lend themselves to being like, oh yeah, clearly the cutoff needs to go like here or there. Like this all seems to be pretty continuous. So if I put my line here or here, I didn't really see that I was going to do anything objectively reasonable, so I just left them where they started. Also because like this was for homework and not for publication, so I didn't have an informed opinion on what I wanted to see and show.
Even when you go to publish it, it's also probably okay if you don't have like perfectly rationalized cutoffs either.
Yeah, I mean, I really don't. I mean, this is a what, 10 to the minus 5 adjusted p-value. All right, yeah, that sounds about right. I don't know. So yeah, there was nothing there that I wasn't like cutting across any populations that looked weird, so I just left it alone. So this is all the genes, right? So then I select this is some data wrangling where I select my significantly differential expressed genes. So I take my, well, actually up here I did the annotation where I grabbed the Homo sapiens database. I used my row names, which is the ensemble IDs, as keys. So the keys is row names, the key type is ensemble. And I grabbed the ensemble entres, the gene symbol and gene name from the database, and then did a left join by the ensemble ID to add the gene symbols to my results table. And then from that annotated results table, I filter out everything that has a p-adjusted value of grade smaller than 0.05. I sort it by that p-adjusted value, and I write it into 2 CSV files. The only reason I use the write CSV2 is because this is going to be a semicolon delimited file and not a comma delimited file, because my computer is European and Europe uses semicolons to delimit their files because we use commas for decimal points. And I don't feel like messing with that, so I just have it write 2 files. Right, so the next thing I'm trying to do here is going to be heat maps of gene expression of all the overexpressed genes. So this is what that looks like. It's glorious. So I take my count matrix, so from the VSD. So this is the normalized counts, not the raw counts. So I just take the raw counts, and then I create 2 indices. I create an ensemble ID index and a gene symbol ID index. And basically, I grab the symbol and ensemble columns from my annotated significant results table. So what these 2 indices are is a list, or a vector, I suppose, of ensemble IDs for all of my significantly differentially expressed genes. So I then subset the matrix of like my complete count matrix with the ensemble ID index. So basically now I only have the count matrix for my significantly differentially expressed genes. And I turn the row names of that count matrix into the gene symbol, and then grab the top 20 lists for that. So this here is just what I want to annotate my heat map with. So this line says that I want this little color bar up here that says condition, but I want this to appear. And then I make 2 heat maps. The first one is what you're seeing right now, where I plot all the differentially expressed genes based on the count matrix that I made up here. I plot them all, and I annotate it with the condition from the metadata. So if we zoom in, any day now. Yeah, we again see that the PDs and controls don't cluster mutually exclusively. There's controls intermixed with the PDs, but they do group kind of together. So you see the dendrogram on the side here again. These are in the bottoms here. From the bottom here, these are the sample names. But you do see that there's sort of like some signatures going on where the data kind of separates. So like you see a bit of an enrichment of higher expressions over here and down here and lower here and here. But I mean, with this number of genes, like there's not that much you can tell from this heat map. So then when we look at the next one, this heat map here is where I used this top 20 matrix I created up here, where it's only the top 20 differentially expressed genes. So if we zoom into this, again, samples don't cluster perfectly, but we can look at what the heat maps looks like for the top 20 genes. Now, there's a few NAs here, and that's because these were ensemble IDs that didn't map to a gene symbol in the annotation database. And that's because when I Googled a couple of these ensemble IDs, it's because they mapped to pseudogenes and stuff. I wasn't really sure if it was kosher to just kick those out, because I kind of wanted to, but I just kind of left them in for now. Yeah, I don't know if you have an opinion on that, Henry. If it's kosher to just be like, no pseudogenes, please.
I mean, at the end of the day, like it's really only worth reporting what you can interpret.
Yeah.
So if it's not interpretable, then it's probably not worth reporting, at least in like a figure. You can always include it in like a supplemental table, of course.
Yeah. Yeah, that's kind of what I thought. But I just, when in doubt, I just kind of left it in for completeness sake. But yeah. All right, so this was everything up till last week. So what's coming now is the overrepresentation analysis and the GSEA. So here I make a vector of overexpressed gene symbols. So I take my annotated significant results table that only contains significantly differentially expressed genes and has the annotation. I filter them by anything that had a log fold change of greater than 2. So these are overexpressed now, not underexpressed. And I pull only the symbol row or the symbol column. So the symbols, the gene symbols of that as a vector. And then I go the MSI and the the macular signature database that contains the gene sets. I specify the species Homo sapiens and category C5, which is gene ontology database. And so I grab this and then select only the gene set name and the gene symbols associated with each gene set from that. So if we look at gene sets, where is it? Nowhere. That's interesting. Oh, no, here it is. Yeah, so it'll have the gene set name, so oligoglucan phosphorylase activity, and then the gene symbols associated with that set. So these, whatever, 10 genes here are apparently involved, or not sorry, the first 6 here are apparently involved with this biological process. So I then run the enricher function that will look for overrepresentation. And so the genes that I want to search through are my overexpressed genes that I specified up here from my DSeq analysis. And the term to genes are sort of the mapping of what I want to do with those gene names, those gene symbols I pass it, is the gene sets data frame here that contains gene symbols, which is what I'm passing it here, and it contains the Go terms that go with those gene symbols. I turn the results into a data frame and save them as CSV files because I can, and then I do my plots here. So this is the one that makes sense to me. These are the results that make sense to me. The ones that you'll see in the future make a lot less sense because this is Parkinson's disease. Parkinson's disease is a proteopathic disease, so that has a lot to do with aggregation and misfolded protein, and everything here has to do with protein folding. There's de novo protein folding, there's protein folding, there's topologically incorrect protein, there's chaperones, there's all manner of anything to do with when something goes wrong with proteins is on here, which totally makes sense to me. Inclusion body assembly, I don't know, this might have something to do with trying to clear aggregating protein, but the counts here, I assume this is how many genes mapped to that Go term in my overexpressed genes. So they're not very high, so it's only like a few genes per Go term, which I don't know if that's worrisome or not, but it is what it is. And when we look at the bar version of that, again, we see everything to do with protein folding. So this made me happy. Then I did the same thing with the underexpressed genes, which I assume you can do just as well as you can with overexpressed. I don't know that that would be somehow wrong. I don't know. It's been a while since I've dealt with Parkinson's disease, but nothing jumped out at me that was like super fascinating.
So Simon, sorry, I don't mean to interrupt, but
No,
can you take another look at line 151?
Oh no, what'd I do? Wait, yeah, smaller than 2. Okay.
So since you're looking for underexpressed genes, maybe you meant to say smaller than -2.
Oh, I totally did mean to say that, didn't I?
Yeah, I only noticed this so fast because I literally make this mistake once a week.
Okay, well, let's try that again.
Oh, it looks like you only had 25 underexpressed genes.
Where does it say that?
So if you look at the,
Oh,
yeah.
there it goes. Okay, so it's not enough to do anything with.
Yeah, unfortunately, Enricher is one of those, or like any overrepresentation, kind of relies on you having enough genes to do something useful with it.
Yeah, makes sense. Okay, well, then that is moot. Okay, so this was where we were at. Okay, well, forget I said anything about underexpressed genes. They're pointless, at least in my data set. There's not enough of them to do anything with. Which is actually interesting in and of itself, that apparently Parkinson's disease's changes are mainly, the changes in gene expression that it induces are mainly toward higher expression of some things, not so much lower expressions of other things. I'm sure someone who's interested in Parkinson's disease might find that fascinating. Okay, so then I did the GSEA. So the first thing I did is add a GSEA metric to my annotated results data frame. Now, it's important to note that this is the total annotated results, not just the significantly differentially expressed genes. So you just use gene enrichment on only your significant genes, but GSEA on the whole thing. So the first thing I did here is what we learned last time, to replace all the 0 p-adjusted values with the minimum, the smallest number that the machine is willing to handle. Make a new column with the GSEA metric. That's minus log 10 of the p-adjusted value times the sign of the fold change. Filter out anything that's NA, and then arrange it and then sort it in descending order by the new GSEA metric. So if we look at the histogram of GSEA metrics, that's what it looks like. A lot of them not very changed, and then some very changed on either side. So I then extract the vector of GSEA metrics and gene symbols. So this is, I'm pulling only the gene symbols and the GSEA metric, dropping all the rest of the data, and turn it into a 2-column data frame. Run the GSEA analysis and get 1,000,001 things that have to do with immunology for some reason. I don't know why. Like humoral immune response, molecule bacterial origin, T-cell activation. I'm assuming a biotic stimulus is probably an immune challenge of some description. There's some extracellular matrix stuff going on, which that might be interesting. Some glutamatergic synapse going on, but Parkinson's disease is largely a dopaminergic disease, so I don't really know what the glutamatergic synapses have to do with anything. So this was like a bit puzzling, I'm not going to lie. So, I mean, people who are still working on PD will know more about this than I do because I did this for my master's. Surely there's an immunological component to the reaction to Parkinson's disease, but I have trouble believing that it's like the most important changes, that the most important biological changes in Parkinson's disease have to do with immunology. Like, I sense that this might be because the tissue that they used for this study was cortex and not midbrain, which is where the dopaminergic neurons are that are dying. So that if they're seeing something in the cortex, maybe the disease is so late stage that cortical dysfunction is already happening, and there might be a lot of neuroinflammation just because neurons are starting to die left and right. I don't really know, but I would be hesitant to call this a breakthrough finding for Parkinson's disease. What are your thoughts, Henry? You're muted.
Can you go back to your data again?
Yeah, like where?
So if you go down to your, let's see here. Where did you run your GSEA?
Right here.
Oh, there you go. Can you look at your GSEA res _df? So one of the complaints I have about cluster profiler is that I don't actually think their summary plots for GSEA are particularly informative. I personally argue that it's better to sort by NES rather than p-value, because that actually can be a lot more informative with respect to. And so can you sort it one more time? Sorry, because you're doing the bottom first. Yeah. So that I think can actually be a lot more informative with respect to like what's actually being overexpressed or underexpressed.
Mhm.
So I don't know if this is like any more.
It's also a lot of immuno. I mean,
Okay.
the first one is something transcribed mRNA catabolic processes, but bacterial origin, some ribosome stuff, beta T cell, interferon gamma, more beta T cells, interleukin 8 was production. Yeah.
So where are your, if you search in that, do you still have the protein?
Sorry, do I have?
Oh, like your pathway is
Oh.
related to like protein processing and
Yeah,
stuff like that. Yeah.
it's fine.
So let's see where those fall in here, because this is actually a really interesting illustration of how overrepresentation analysis is different, I feel like, from something like GSEA. So yeah, that one doesn't even show
Chaperone's
up.
gone. Sorry? Oh, is this case sensitive?
No, I don't think so.
Nope. Okay, so chaperones are gone. Let's see. Topologically incorrect protein is still there. 1.6, I don't know if that's a lot. What is NES? Like, what's that score?
No, I mean that's not a crazy amount of enrichment. So this is actually really interesting to me, Ben. So I think something to keep in mind here is that when you're interpreting this, you have to consider what the tissue source is. So you mentioned that it's cortex. Is it sorted cells from the cortex, or is it just straight up cortex?
I don't remember if it's sorted cells or not.
Okay.
I think it's probably just bulk cortex.
So something that I saw with the Alzheimer's data that I processed was that the top thing was immune response. And I suspect that's because I was dealing with whole tissue, like whole heterogeneous tissue, that genuinely was undergoing an increase in like microglia activation and that sort of thing.
Yeah, that's what I suspected too. That it's just a response to the late stage widespread death that's going on.
Right. And the other issue is that you have what I would consider to be a survivorship bias. So the way that bulk RNA-seq works, you could actually be seeing a signature that's more indicative of what has not died,
Yeah,
rather than what's actually being overexpressed.
like neuroprotective things.
Right. Exactly.
Yeah, it's like that those World War II planes with the bullet holes where they figured out the. I don't know if you've seen those. There's like schematics of World War II planes that came back and they were just riddled with bullet holes in various places, and they decided to reinforce the planes not in the places where they had bullet holes, but where they didn't, because those were the places where if the planes got hit, they didn't make it home.
That's really smart. Wow.
Yeah, survivorship bias, like, yeah.
So I think this was actually, so I mean, first of all, like really well done. Like this is awesome work. I think this is actually just such a super interesting illustration of so much of what we talked about, but also why at some level it's probably important to move to a single cell system when you're dealing with a tissue like this, because I think it's so hard to interpret from the results you're seeing. It's just very hard to interpret what's due to genuine changes in expression and what's due to changes in tissue composition. Because if you look, I saw you had this G0S2 gene. Isn't that's related to cell cycle as far as I know. And so that might be indicating that you have a larger proportion of cycling cells in one sample versus another, which might mean you have more glia in one sample versus another because of neuronal death. Or, or it could mean cell cycle reactivation. Who knows, right? So that's why I think it's important to move to a single cell model at some point. But this is really awesome, and I really appreciate you sharing this.
Yeah, no problem.
So do you have any, I guess, like kind of other thoughts or like conclusions that you drew while doing this?
Yeah, so I actually did the GSEA again with a different GSEA metric. I just used the log2 fold change as opposed to the p-value with the sign of the log2 fold change to like look at ranking genes by how much they change rather than how sure we are that they change.
Sure.
And the results were very similar. So nothing revolutionary happened there. I just gave it a try. So I think one of my takeaways was that your choice of analysis really comes down to the question you're asking and like what you want to see. So it's not that GSEA is just better. Like I think in my case, I'm pretty like I would bet a lot of money that the overrepresentation analysis gave more true results of what Parkinson's disease does to a cell than the GSEA.
Mm-hmm.
But that you have to like really think about where your tissue comes from and how they processed it. And like I really feel for all the bioinformaticists that want to scream every time a biologist comes to them with already done experiments and wants them to answer questions, and they have to explain to them that they needed to have this conversation 6 months ago. Because like, yeah, depending on what tissue you use and if you sort it, if you don't sort it, and what kind of, how you do the biology of your experiment, the things that your bioinformatics analysis will tell you will just differ drastically, right? So like
Exactly.
taking cortex from a late stage Parkinson's disease patient will answer very different questions than taking midbrain from an early stage Parkinson's disease patient where the like substantia nigra is still like in the process of dying.
Yeah, I couldn't agree more. That is a feeling that I feel a lot of the time. But that's why you're there, because then you can, then you can explain it, and then they can design the experiments in a way that will produce interpretable data.
Yep. All right, that is all I got.
All right, really great work. And so let's go ahead and break now. So actually, I should just say before we break, does anybody have any questions or any final comments they wanted to make? I think we might have lost a few people while we went a little bit over time there.
Yeah, and I had to I went pretty fast. Yeah.
But it's good. It's good that actually you go through the homework.
Yeah.
So I know the next week are going to be the Linux shell.
Right. Yeah. So Dr. Fang Gong is going to do sort of an introduction to Linux shell next week.
Okay. Yeah. Yeah. But so which one? Just a logistic thing. Which one that we need the cloud one? It's not after next week.
So that's going to be next week. We'll be using the cloud computers for next week.
Oh, okay.
Yeah. We'll get those. We'll get those set up. I mean, a worst case scenario, we'll just have everyone log into DataCamp and use the Linux shell in DataCamp. But hopefully we don't have to do that. Hopefully we can set it up.
We got our one. Yeah.
Yeah.
All right. Good. Just.
All right.
I'm gonna sign off. Okay. Thank you.
All right. Thank you all so much. See you next week.
Bye-bye.